changes cannot be taken place in the vaccine. That is a major characteristic, and should provoke the vaccine must provoke the immune system for the long lasting time. Okay, for the long time, it must provoke the immune system to produce the antibodies against the specific antigens, and also T cells against the antigens, specific antigens, whatever for what which purpose we are taking vaccines, or persons given the vaccines, that antigens, for that antigens, antibodies must be produced for the long time, and T cells, active T cells production will be stimulated for the long time. That is the major characteristics of the vaccine. Okay, and especially the coming to the history, and in the year of 1798, smallpox vaccine uh, has came into the picture after some smallpox, rabies, plague, diphtheria, uh, pertussis, and tuberculosis, tetanus, yellow fever, and different types of vaccines are came into the picture after smallpox. And immunity to infectious diseases can be achieved by active or passive immunization. Okay, immunity to infectious diseases in general achieved by active or passive immunization. Okay, in general or by giving the vaccination. In active immunization, what they do? The immune system is stimulated to develop its own immunity against the pathogen. Okay, in what happens in the active immunization in general, the immune system is stimulated. Okay, whenever the antigens or pathogens invaded into our body, our immune system is sensitized, and after sensitization of our immune system, it produces different types of active immune magic bullets. That means antibodies are also called as magic bullets. Okay, different types of classes of antibodies are produced after active immunization, and different types of cells also get active sensitized and activated after invading of the different pathogens. This is active uh, immunization. And what happens in the active immunization, how it is achieved? Inoculation with microbial pathogens. Okay, whenever the healthy person inoculated with the uh, active bacteria or different types of pathogens, that induces our immunity but do not cause any disease. Okay, for this uh, it is a vaccine. Okay, vaccine first, active immunization or active vaccination process should be done by the giving microbial pathogens that induce immunity. Okay, whatever we are giving the um, vaccines we are giving, it sensitizes our immunity but not create any illness in the giving in the taking persons. Okay, vaccine means it produces or it evoke the immune system but not affect the person by giving the diseases. Okay, and with antigenic components from the pathogens, the achieved can be achieved. Active immunization can be achieved by two ways. First way is that giving the pathogens directly to the person in general, or giving the antigenic compounds. Okay, productions. Okay, products of pathogens should be given as a vaccines. Okay, should be utilized as a active immunization or active as vaccines. Okay, this is about active immunization or active vaccination. Coming to the passive immunity, or as our friend said in the starting class. Okay, antigens may be used as a vaccines. Otherwise, we can use antibodies, produced antibodies as a vaccines. Okay, as she told, and two types of vaccines are there: active vaccines and passive vaccines are there. In the active vaccines, what they do? They inject the antigens directly, or antigens in the sense pathogens directly. The procedure is there. They do not give the directly active and pathogens, but they inactivate the pathogens. After inactivation, they give as a vaccine. Okay, the process is there. I will discuss later. But two types of vaccines are there: active vaccines, and passive vaccines. Okay, passive immunity. Okay, immunization. And what happened? What they do in the passive immunization? In the passive immunization, they directly give the antibodies. Okay, what which previously produced in the synthesized in the diseased persons. Okay, previously there in the infected persons, they collect the antibodies from the infected persons and give the patients. Okay, after first exposure in the persons, antibodies are produced in general, and they collect those antibodies from the previously affected persons, and they use as a vaccines for treating the patients, okay, who are suffering from the disease or illness. Okay, that is passive immunization. They are in the passive immunization. What they are doing, they are giving the antibodies instead of the pathogens or pathogen products. In the active immunization, they are directly giving the pathogens. Means inactive pathogens are killed pathogens or pathogens products. For example, bacterial products. Okay, bacterial proteins, bacterial enzymes. Okay, bacterial products. Okay, in the active immunization. Okay, these are the types of immunizations. Okay, vaccines. Okay, different types of categories of vaccines are there. Okay, different classes of vaccines are there. Okay, uh, basically two types: active immunization or active vaccines. Okay, passive vaccines. Okay, and vaccines are classified following different types. Six categories are given in the your book, but there are many types of vaccines are there. First of all, bacterial vaccines. Okay, in the bacterial vaccines, two types of vaccines are there. Okay, different types of bacterial diseases, and vaccine having live but attenuated bacterial cells. Okay, vaccines are live but attenuated bacteria. Okay, live attenuated. They are bacteria. Disease causing agents are being given, are being inoculated into the patients, but after inactivation of the pathogens, they are giving after inactivation or after weakened. Okay, after weakened by treating with the different types of procedures, uh, after treating with different types of procedures like physical treatment, like chemical treatment. After treatment, they are not fit to produce any diseases, but they are fit to evoke our immune system, but they not create any they not they didn't they do not cause any diseases like in general infection. In the vaccine, they are giving weakened or inactivated bacteria. They are using inactivated bacteria for the vaccines. For example, BCG, okay, anthrax, they are giving direct bacterial, inactivated bacterial, okay, weakened bacteria. Plague, in the plague also, they are giving inactivated or weakened bacteria. And Brucella, they are giving vaccine, they are using vaccines after Brucella bacteria weakened after inactivation, they are giving as a vaccine. And in the bacterial vaccines, they are also giving dead cells. Okay, dead bacterial cells also giving. They are dead, but they cause a disease. They did not cause any. They do not cause any. They do not produce any disease, but they evoke our immune system to produce antibodies against the antigens on the surface of dead cell, a dead bacteria. Are you understand, Amma? Hello.
Yes, sir.
Yeah. They are bacterial vaccines are two types. What they are doing in the first type? They are giving live but attenuated. But attenuated means weakened or inactivated. The inactivated bacteria are giving uh, for the BCG disease and anthrax disease, plague disease, brucella disease, and different types of diseases. They are giving inactivated bacteria. Okay, inactivated means they treating bacteria are treating with the different types of physical agents and chemical agents. Okay, after weakening of the bacteria, they are using as a vaccines for the different diseases. For example, BCG, anthrax, plague, brucella. In case of typhoid and cholera, they are giving dead cells. Okay, they are also using cell but dead cell. In case of other BCG, they are giving live but not fit to for the not fit for disease making. Okay, that is the difference between live and dead vaccines. Okay, and we are also having viral vaccines. Viral vaccines also two types. Vaccines having live virus. Okay, for example, MMR, measles, mumps, rubella virus vaccine. Vaccine is a live vaccine, and smallpox vaccine also live virus vaccine. Influenza, Sabin, poliomyelitis virus, and Covishield virus also vaccine also live virus vaccine. Covishield for COVID-19. Okay, uh, some of you also have taken Covishield vaccine for COVID-19. So that is live virus. Okay, live attenuated virus. And next, some of the vaccines are killed viruses. Okay, killed viruses are acting as a vaccines. For example, shock vaccine, poliomyelitis vaccine, and very important vaccine is that Covaxin. Okay, for the COVID-19, most uh, listened vaccines are most of times listened we have heard about two types of vaccines. They are Covishield and Covaxin. Covishield is a live virus, a live attenuated virus vaccine. And coming to the Covaxin, killed virus. Okay, killed virus is a virus vaccine is a Covaxin or inactivated killed vaccine. Okay, this is the difference between Covaxin and Covishield. In the viral vaccine also, there are two types of vaccines are there. And coming to the inactivated vaccines, what exactly meaning of the inactivated vaccine is that in the what they are doing in this process, the ability to cause disease has been destroyed or weakened. Okay, what they do in the inactivated vaccines synthesis or production, the ability to cause disease. Okay, which molecules are causing the diseases, those molecules are uh, eliminated or destroyed, or those structures are destroyed or weakened in this process. After the destroying or weakening, they use this as a vaccines in the inactivated vaccines. How they produce these type of vaccines? The type of vaccine consists of virus, bacteria, particles that have been chemically treated with formalin. Okay, how they are producing? By treating with formalin, they inactivating the active sites on the viruses or bacteria. What happening after the treating with the formalin? Formalin inactivated the different types of molecules. Okay, I will uh, I will explain in detail. And physically treated with the heat or eradications so that they are no longer able to replicate. Okay, and such vaccines are called as killed or inactivated virus or bacteria vaccines, and possesses antigens common to the original pathogen but do not replicate. This is a very important point. In what happens in the inactivated vaccines, the uh, replication capacity will be uh, destroyed. Okay, replication capacity will be destroyed, and then the causing agents on the surface of the virus or bacteria also be destroyed in this type of vaccines. They do not replicate in the body of the person. Okay, whenever we take the this type of inactivated vaccines, vaccine uh, containing the bacteria virus, they do not replicate in general or in general as is in general viruses or bacteria. Okay, they do not replicate in case of inactivated vaccines. And for example, there are two types: inactivated bacterial virus vaccines and inactivated viral vaccines. Okay, and for example, inactivated bacterial vaccines are pertussis vaccines prepared with killed uh, pertussis bacteria, and typhoid vaccines also killed from the Salmonella typhi, and cholera also acting as a vaccine and prepared by this process, the inactivation of the bacteria or killing of the bacteria after killing of the Vibrio cholera, they are using as a vaccines. Okay, for example, viral examples: polio vaccine. Okay, inactivated or killed polio vaccines are giving as a vaccine, and influenza vaccine also killed or inactivated vaccine by treating with the formalin. Rabies vaccine also killed viral vaccine. Okay, these are the few examples for the inactivated vaccine. Okay, and what are attenuated vaccines? Okay, attenuated means mutated. Okay, mutated means in this what they doing, they lose their ability to cause significant disease but retain the capacity for multiplication. This is very important. They multiply in the person but they do not cause any disease. Okay, they are having the ability to multiply after after the giving the vaccine, after getting into the person, but they do not cause any disease. Okay, disease causative agents are eliminated or deleted from the sequence or is from the surface of the bacteria or viral particle, but replication ability is retained. Okay, after taking the after entering into the person, they normally in general actively they proliferate or multiply in the person. Okay, in case of inactivated, they are deleted their multiplication capacity in inactivated vaccines. But in this type of attenuated vaccines, multiplication ability is retained, but the disease causative ability is deleted in case of this two both. In case of both, ability to disease causative agents uh, will be deleted or deleted in both vaccines, but ability to multiplication in the body is retained in the attenuated vaccines, but not in the in case of inactivated vaccines. Okay, this is the main difference between two uh, vaccines. Okay, attenuation can be achieved by growing a pathogenic virus or bacterium for prolonged period under the abnormal culture conditions. Okay, under the abnormal culture conditions, they lost its ability to cause the diseases, but it it has it retains its ability to proliferation. And most of the viral vaccines used nowadays are live attenuated vaccines. Okay, most of the vaccines are live attenuated. What happening in the live attenuated? They are multiplying in the person, but not cause any diseases. Okay, that is the specialty. That is the beauty of the vaccine, attenuated vaccines. Vaccines made in this way are often the most successful vaccines, probably because they multiply within the host, thereby causing a large immune response. Okay, they are continuously multiply in our in the taking person, whoever taking these type of vaccines, those in those persons, multiplication process is going on from the taking period, from the taking time, but they do not cause any. do not create any disease symptoms in the uh, persons, okay, whoever taking the vaccines. And however, these attenuated vaccines also carry greatest risk because they can mutate back to the virulent from any time. Okay, we scientists are making these types of vaccines by mutating the gene sequence. Okay, where exactly disease causative sequence is there, the scientist recognizes those sites, disease causative sites on the on the DNA molecule. They destructs or they manipulate the sequence of the disease causative agents of the DNA. After mutation, they lose their property of the disease making disease causing in the persons, but retaining the uh, multiplication, they lose the property of causing the disease. Okay, that is the beauty. And next, such mutations, okay, they are doing mutations in the sequence of the pathogens or viral or bacterial sequence. But sometimes what happens that it results in the induction of the disease rather production against it. Sometimes what happens, they retains. Okay, great risk is that they can mutate back. Okay, after some time. Okay, we cannot expect the time how much time it take, but after some time it may takes 1 year, 10 years, 50 years, 100 years sometimes, but it it can mutate back to the virulent form at any time. At that time, person health will be person get diseases. Okay, a person get disease after uh, mutation, back mutation. Okay, mutation, back mutation. That is one of the limitation for this attenuated vaccines, but they are successful vaccines among the different types of vaccines. And for example, mutant typhoid vaccine, a vaccine for whooping cough, BCG vaccine, yellow fever vaccine. Different types of vaccines are made from this method. Okay, attenuated method. And we have toxoid vaccines. Okay, toxoid vaccines are also being made with the by using different technologies, and can be made by purifying the bacterial exotoxin and then inactivating the toxin with the formaldehyde to form a toxide. Okay, what they are doing in this type of vaccine making, they extract the exotoxin from, especially from the bacteria. After getting into the system, bacteria produces some chemicals. Okay, some secretes produces some chemicals to protect them from self from the our body immune system or body enzymes, body cells. In this process, they produces some chemicals. We can call it as exotoxins. Okay, exotoxins of bacteria. So scientists collect the exotoxins from the bacteria exactly in the culture, and they use as and after collection of the exotoxin, they processes the toxins, exotoxins. That means they treat with the formaldehyde or formalin, and after treating with the formalin, they get inactivated, and the property of disease causing will be lost after treating with the formaldehyde. So after inactivation of the exotoxins, they directly give the they directly use as a vaccines. This is one type of the vaccine making. Okay, this is also uh, being used in the different types of diseases. Okay, and what are the limitations? Limitations for treating the toxin with the formaldehyde denatures the protein, so that is no longer dangerous, but retains some epitopes. Okay, this is a procedure exactly, but retains some epitopes in the molecule that will elicit protective antibodies. Okay, after the treating with the formaldehyde, they denature, but some capacity of the eliciting immune system will be retained after the treating with the formaldehyde. By using these inactivated exotoxins as a vaccine, they produces they evoke the our immune system, because they are also having the capacity to stimulate our immune system, and produces antibodies and T cells against this exotoxin, and uses as a uh, vaccine. Okay, also working as a vaccine for this uh, toxoids. Okay, toxoid vaccine, this is about toxoid vaccine. But some of the difficulty of obtaining sufficient quantities of purified macromolecules of bacteria is very typical task. Okay, this is a simple talking about the toxoid or any vaccine is a simple task, but making or production in the laboratory is the extensive work and very typical task to produce vaccines in the laboratory. And difficulty in the making the toxoid vaccines is that purification of exotoxins from bacteria is a very big task, and that is one of the limitation. And to overcome by cloning, to overcome it by cloning is a, of the exotoxins genes is a very uh, easy to easier than the getting exotoxins from the live bacteria. It is easier to make exotoxins by using genes. Okay, in the cloning. And easy grown host cell. Okay, these are the genes inoculated with the host cell, and after inoculation of the exotoxin genes into the host cell, they produces exotoxins in the host cell. After the production of the exotoxins, we collect the exotoxins and use as a toxins after treating with after treating with formaldehyde. This is about toxoid vaccines. Okay, and we have another type of vaccines. Those are recombinant. Okay, recombinant antigen vaccines. Okay, the very important vaccines among them. And in this process, the gene encoding any protein is separated first. Okay, whatever it may be, gene encoding with antigens. Okay, gene encodes antigens on the surface or anything on the pathogens or in the pathogens are recognized and separated. Okay, cloned and expressed in the bacterial and mammalian or yeast cells using recombinant DNA technology. The expressed antigen is used for vaccine development. Okay, after getting the genes from the bacteria or viruses, the gene from the bacteria viruses are inoculated by using recombinant DNA technology into the host cell or vectors. And after the inoculation, they produces antibodies or antigens along with the general DNA transcription translation, and the produced antigens or products will be collected, and they used those produced are antigens used as a vaccines in this type of technology. The best example of recombinant vaccine is hepatitis B vaccine, and yeast cell are grown in the large fermenters. Okay, in the yeast cell, the hepatitis B virus gene will be inoculated into the uh, in the yeast cell. In the yeast cell, along with the yeast DNA, this hepatitis B uh, gene inserted and translated and transcripted and translated. And after production of the proteins, the proteins are collected and used as a vaccines in the technology in this technology. And among them, for example, few of them vaccines, recombinant vaccines, diphtheria, tetanus, bacteria, virus vaccines, parathyroid fever, typhus fever, Okay, different types of bacterial vaccines are made by using recombinant antigen technology, vaccine technology, and types of vaccines are produced for the uh, taxoids. Okay, okay, all these are taxoids. Diphtheria taxoid are produced by using recombinant DNA technology. Tetanus toxoid also used, also produced and used as a vaccine in this recombinant technology. And different types of uh, disease making genes are are used as a recombinant antigens. Okay, in this technology. Okay, different types of vaccines are given in your page number in the book in the page number 192. The different types of vaccines, recombinant vaccines, okay, recombinant different types of technology vaccines are there. Plague, tuberculosis, typhoid, okay, meningitis, different types of viruses, yellow fever virus, disease, measles, mumps, rubella, polio. The different types of vaccines are made by using recombinant DNA technology. This is very, very recent and advanced technology. And we have another vaccines class, that is recombinant DNA vaccine. The recombinant DNA vaccine, recombinant DNA, in the what they do. The principal DNA vaccine production is to use DNA encoding the antigen of the pathogen as a vaccine. Direct DNA, okay, direct DNA is used in this type of pathogen and recombinant DNA technology. Direct DNA encoding the antigen of the pathogen as a vaccine. In this procedure, a virulent virus or bacteria is created, which can be used as a vector vaccine. A virulent means non-pathogenic. They do not make any disease. Okay, they do not make any disease symptoms in the person. Okay, the number of organisms have been used for vector vaccine, including vaccine virus, vaccinia virus, attenuated polio virus, adenovirus. Different types of viruses are used and instilled into the plasmid under the control of the promoter of the vaccinia, uh, thymidine kinase gene. Okay, in the page number 193, they given one procedure to prepare the recombinant DNA vaccine. Okay, recombinant, how they prepare, how they produce recombinant DNA vaccine. They have given as a one explanation or figure. So what they do, they uses plasmid DNA to make the recombinant vaccine. Okay, uh, what happens in the case of plasmid DNA? In the plasmid DNA, the promoter is there. Okay, promoter is there. Adjacent to promoter, they cut the DNA sequence adjacent to the promoter. Both side of the promoter, TK gene. TK gene means thymidine kinase gene is there. In between these TK genes, vaccinia promoter gene is there. Or promoter sequence is there. Adjacent to promoter gene, they cut the DNA with the restriction enzyme. Okay, they cleave the site, and at that cleaving site, our interested DNA encoding the pathogen gene will be inserted after the cutting the plasmid DNA sequence by using restriction enzyme. And after the cleavage, they do ligation with the enzyme ligase. Okay, by using ligase enzyme, they they uh, they connect, they binds, they uh, uh, link the genes, the DNA cutted DNA will be joined with the help of ligase enzyme. After the ligation, the recombinant DNA or recombinant plasmid will be formed. Okay, which is having the gene from the pathogen. Okay, also it also having the TK gene in between the gene of pathogen, and it transfuses into the culture. Okay, culturing cells, it transfuses into the culture cell. It inoculated into the transfixion will be is one of the another step on the tissue culture. The transfixion will be seen, and after infection, homologous recombination will be occurred, but they give BUDR. Okay, they perform the BUDR selection. What is the meaning of BUDR? They add bromodeoxyuridine, okay, which kills the TK plus cells. Okay, TK plus cells will be eliminated after uh, giving the this BUDR treatment, and only TK minus genes which are having the recombination DNA, okay, exactly pathogen gene, they retain in the culture. The remaining will be eliminated after treating with the bromo deoxyuridine. So, and this recombinant technology has revolutionized, revolutionized, revolutionized in the field of vaccinology. This is a simple procedure. You might not understand in one time, but uh, you understand. I will, if you want, I will send a video of the recombinant DNA technology, recombinant DNA virus. Uh, Take my number and just text me in the WhatsApp for the video. I will send videos of this vaccine. Okay, recombinant, especially you can study all those things, but except this recombinant DNA procedure, you might not understand very well for the first time. But after seeing this video, you can understand everything about the recombinant DNA vaccine, how they are producing recombinant DNA vaccines. Okay, I will send the video. Just text me just in the WhatsApp number for the WhatsApp. So this is about the recombinant DNA vaccine, and we are also having DNA vaccines. DNA vaccines also there. With the DNA vaccines, the DNA coding for the specific component of disease causing organism in the injected is injected directly into the muscles of the recipient. Muscles of the recipient, disease caused to DNA, that not recombinant DNA, but direct DNA. Okay, direct DNA of pathogen. In the what happens in case of recombinant DNA, the DNA of the pathogen was inoculated. Okay, in general, they are inoculating the DNA, causing pathogen causing DNA gene into the plasmid DNA. But in case of DNA vaccines, they are giving disease caused to pathogen DNA directly into the muscle of the person or recipient. The DNA is taken up by the muscle cells, and the encoded protein antigen is expressed, leading to the both a humoral and cell mediated responses. Okay, they evoke or stimulate the recipient immune system after some time. That both humoral and cell mediated immune responses are get stimulated, and this is the very important. And the viral DNA is expressed not only by the muscle cells but also by the dendritic cells in the area that taken up by the plasmid DNA and express the viral antigen. Okay, after engulfing uh, the viral particle by the phagocytosis by the dendritic cell, then at that time also the viral antigens expressed on the surface of the dendritic cells. That is one of the another way to uh, evoke our immune system. But they evoke our immune system directly by after giving into the muscle cells. Okay, it goes and directly stimulate the our humoral system, humoral immune branch, and cell mediated immune branch. And DNA vaccines offer advantages over many of the existing vaccines. The encoded protein is expressed in the host its natural form. There is no denaturation. DNA vaccines cause prolonged expression of the antigen which generates a significant immunological memory. Okay, in humoral branch of the immunity, the memory will be attained for the type of DNAs, and proved able to protect against the tuberculosis, SARS, and the smallpox and other intracellular pathogen. This technology is being used for these types of diseases. In addition, more than dozen different DNA vaccines against HIV-1, the cause of AIDS, are in clinical trials. Okay, clinical trials are going on under progress for the AIDS disease also. This is about DNA vaccine. Okay, main difference between DNA vaccine and recombinant DNA vaccine is that in the recombinant DNA vaccine, the pathogen DNA will be, especially pathogen gene will be inoculated into the plasmid or vector. But in the case of DNA vaccines, directly pathogen DNA will be inoculated or injected into the muscle cell of the recipient. That is a very important difference between these two vaccines. And we have another synthetic peptide vaccines. Okay, and another synthetic peptide vaccines also there. Small peptide sequences, okay, corresponding with important epitopes on microbial antigen can be synthesized readily or ready-madely in the laboratory. Okay, what they do in the laboratory, scientists identify the sequence of the peptide, okay, amino acid sequence of the disease causative protein or peptide uh, is identified and can be synthesized. Okay, they identify the sequence and they synthesize artificially in the laboratory by using different techniques and tools and techniques. They manufacture or produce or synthesize ready-madely in the laboratory. Okay, the sequence will be synthesized. Okay, epitope sequence. Okay, disease causing agent on the antigen is called as epitope. Okay, or antibody binding site on the antigen is called as epitope. Okay, that epitope sequence will be synthesized artificially in the laboratory and used as a immunogen, okay, or antigen. And these and immunogens not directly can be used as a vaccines, but it requires uh, 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 adjuvants. Okay, and the T cell dependent carriers are incorporated into the adjuvants. Okay, for immunogenicity. Okay, it cannot generate, it cannot provoke our immune system individually, but it requires additional support as a anti-adjuvants are the additional support for the making the immunogenicity in the person. So by addition of these adjuvants, epitopes, artificially synthesized epitopes also can be used as a vaccines. We can call it as synthetic peptide vaccines. For examples, different types of disease. Not they have not mentioned any disease names, but they have given advantages. Okay, chemically purity safely. Okay, we know the sequence of the peptide or epitope, so chemically safe, and ultimate uh, unlimited source of material. We anytime in the laboratory we make the proteins in the artificial, so need not to bother about the production. Low cost. Okay, production of the peptide sequence is a very uh, important, and it can be produced in the low cost. And sterility, easy storage, and delivery is easy. Different types of advantages are there. Along with the advantages, also having the disadvantages. Need to know the amino acid sequence of T cell and B cell epitopes. Okay, we must know about the epitopes of the T cell and B cell also. We know must know also must be know about the B cell epitopes and T cell epitopes. That is a very big task and requires generation of population of memory TH cell. Okay, generation of the memory TH cell production is required in case of to make the synthetic peptides, and peptides which represent a linear sequence of the primary sequence mimic only part of the determinant. Okay, uh, this is one of the main drawback on the surface of the antigen. The different types of epitopes are there, not one type of. So it is the for the another uh, epitope we cannot it cannot used as a vaccines. Okay, poor immunogenicity. Okay, poor immunogenicity is the main drawback of the synthetic peptide vaccines. It can itself it cannot act evokes the immune system, but it requires the additional support as a adjuvant. Adjuvant support is required for the synthetic peptide vaccines. Coming to the another type of vaccine is that multivalent subunit vaccine. This is also one type of recent advanced technology uh, to make the vaccines. Okay, you may not understand in one time, but I have the video link for the better understand of the this multivalent subunit. Name itself indicated as multivalent. Okay, it has multivalent potency to produce different types of immune mechanisms. Okay, it evokes different types of immune mechanisms by having the different multipotent, okay, multivalent potency. Okay, the methodology consists of preparation of solid matrix. Okay, I will share video for your reference. Just go through video. And last one is that plant-based vaccines. Okay, plant-based vaccines. This involves in the development of transgenic plants that expresses not for these plant-based vaccines, not for the not only for the animals, okay, livestock, but also useful for the human beings. Okay, how they are producing these type of vaccines? They producing this type of vaccines by using making the transgenic plants. Okay, making transgenic plants, they inoculating the different types of antigens into the plant genes. Okay, how they are producing, I will explain how they are producing. Development of transgenic plants that express surface proteins of virus that are pathogenic to animals or humans. Okay, those those antigens are inoculated into the plant genes. Okay, for example, agriculture genetics in England has genetically engineered the cowpea mosaic virus to include a surface antigen from the foot and mouth disease virus. This virus affects the livestock in general. Okay, we know the foot and mouth disease about about this disease. This genetically engineered virus. Okay, this genetically engineered virus is used to infect the natural host. Okay, black-eyed pea, and the introduced gene from the foot and mouth disease virus was expressed, okay, handsomely in the plant. Okay, inoculating or infecting the plant with the uh, viral infection, that is especially disease causative virus. With the help with the disease causative virus, plant infection will be made. After infection, plant particles, okay, plant particles is given as a food for the livestock or any other animals. Okay, cowpea mosaic virus eventually kill the virus plant, and therefore the plant needs to be sacrificed a few weeks after infection. One leaf from an infected pea plant produces enough surface antigens to serve as a vaccine. Okay, we in the plant they the proliferation of the vaccine Okay, multiplication of the viruses will be seen after inoculation or after infection, and those plant parts can be used as the vaccines. In the future, scientists are going to plan for producing different types of vaccines, though inoculated vaccines. Okay, inoculation of the fruit, inoculation of the vegetables, inoculation of the different plant materials what we are eating today. Okay, scientists are thinking about that. Okay, this method naturally led to the way to the development of edible vaccines. Okay, edible vaccines, they are not came into the picture, but in future scientists are thinking about the edible vaccines. Okay, edible vaccines that makes vaccination programs are common as eating a banana. In future, we uh, future these vaccines are come in future. We cannot take as an injection. Just we after eating banana, it is enough for the any type of diseases. Okay, that is in future they are thinking about this. The munching of alfalfa sprouts will also act as a anti vaccine. Genetically transported transformed banana is a favorite for developing edible vaccine, because edible vaccine would not need purification, unlike strict refrigeration and injections that make conventional vaccine expensive. And the use of edible vaccines may encourage preventive medicines in the third world, not in present. Third world means in the future. Tomatoes and lettuce have now been transformed to produce hepatitis B proteins isolated from the infected humans. Okay, they are experiments are under progress. All these type of plant-based vaccines experiments are under progress. Edible vaccines is successful. If it is successful, will yield a low tech production, low cost vaccine for many disease in financially challenged countries. Okay, uh, in future, all these vaccines may be replaced by the plant-based vaccines. Okay, we can also call as edible vaccines. Okay, edible vaccines means eatable vaccines. After eating, they are acts as a vaccine. They are acts like a vaccines in the future. Okay, under experiments are going on. Okay, this is about vaccine topic. If you have any doubts, you can ask now. Any doubts, Amma? Hello?
No,
Hello?
yes, sir.
If you have any doubts, you can ask. Memang doubts ఉంటే అడగండి అమ్మా.
సార్ ఇప్పుడు
వ్యాక్సిన్ టాపిక్
ఎస్ సార్ ఏం లేదు
తెలియదు.
సార్.
చెప్పండి
సార్
అమ్మా,
ఇప్పుడు
చెప్పండి.
ఎపిటోప్స్ అనేవి మనకి చేంజ్ అవుతూనే ఉంటాయి కదా సార్. అది వైరస్ అంటే ఇప్పుడు ఇది ట్రాన్స్జెనిక్ కదా.
అవునమ్మా,
ఇప్పుడు ఎపిటోప్స్ అనేవి చేంజ్ అవుతూనే ఉంటాయి కదా సార్.
అవునవును.
ఓకే సార్, అంతే సార్.
ఎపిటోప్స్ అనేవి చేంజ్ అవుతూ ఉంటాయి. కొన్ని ఇన్ కేస్ ఆఫ్ వైరస్ పార్టికల్ అంతే. బ్యాక్టీరియాస్ మాత్రము అంతే ఉంటాయి.
అచ్చా, బ్యాక్టీరియాకి అయితే అదే ఉంటుంది.
బ్యాక్టీరియాలో ఉన్న సీక్వెన్స్ మ్యూటేషన్ కావడానికి చాలా టైం పడుతుంది. కాబట్టి
ఓకే
బ్యాక్టీరియా
సార్.
ద modifications and mutations in the bacterial epitopes takes lot of long time. Okay, don't get అంటే మనం బాధపడాల్సిన అవసరం లేదు, మనం భయపడాల్సిన అవసరం లేదు. బ్యాక్టీరియా ఇన్ కేస్ ఆఫ్ బ్యాక్టీరియల్ డిసీజెస్. ఓకే, బ్యాక్టీరియాలకు డిఫరెంట్ టైప్స్ ఆఫ్ డిసీజెస్ కు మనం వ్యాక్సిన్ తీసుకుంటే మళ్ళా ఒక ఎపిటోప్ తీసుకున్న వ్యాక్సిన్ ఇంకో ఎపిటోప్ కు పనిచేస్తా లేదా అని భయపడాల్సిన అవసరం లేదు బ్యాక్టీరియా కేసులో. కానీ వైరల్ కేసులో మాత్రం కొన్ని ఎక్సెప్షన్స్ ఉన్నాయి. ఈ కరోనా లాంటి ఇన్ఫ్లుయెన్జా లాంటి వైరస్ మాత్రమే ఎపిటోప్స్ అనేవి చేంజ్ అవుతుంటాయి. నాట్ ఫర్ ఆల్ viruses, except few viruses having the capacity to change, like AIDS virus, okay, influenza virus, okay, COVID virus. ఎపిటోప్స్ అనేవి కంటిన్యూస్ గా చేంజ్ అవుతున్నాయి, ఎందుకంటే సీక్వెన్స్ చేంజ్ అవుతూ ఉంటుంది. అదర్ Do not changes its epitopes. Okay.
Okay, sir.
Very good. Manchi question amma. Next. ఇంకేమైనా క్వశ్చన్స్? రేపు ఏం క్లాస్ ఏం డిస్కస్ చేద్దామమ్మా రేపు? మీ ఇష్టమైన టాపిక్. వీ హ్యావ్ immunity to infectious diseases, immunity to parasitic diseases in block 3, and we have tumor immunology, immunodiagnosis and applications in last block. Okay, your wish. Okay,
Tumor immunology, sir.
Tumor immunology discuss చేద్దామమ్మా?
Yes, sir.
Okay అమ్మా, we will discuss tumor immunology.
Sir, epitopes are present on antigen surface, no sir, actually?
ఏమమ్మా epitopes?
Epitopes are present on antigen surface, no sir?
Antigens, always antigens.
Page number 194 in synthetic peptide vaccines.
194.
194.
Amma.
Third line, construction of these synthetic peptides for use as vaccines requires an understanding of the nature of T cell and B cell epitopes. What is the meaning of it, sir?
T cell, B cells also having the epitopes. Okay, T cell where exactly it binding with the antigens. Okay,
sir,
no
but
need to know.
We have the antibodies, paratopes, no sir?
Paratope.
Antibodies.
These are not antibodies. These are cells.
Okay.
Cells are having epitopes. On the surface of antibody only paratopes.
Okay, sir.
Also on the surface of T cells, B cells also having epitopes.
Okay.
Okay,
sir.
Okay.
Sir, please share your number, sir.
Okay, ma. Write down. You don't mind.
Sir.
89
89
85
85
70
70
63
63
64.
64.
Dr. Siddhaya.
Siddhaya, sir. Okay.
If you have any doubts, share in text me in the WhatsApp. I will respond. Okay, quickly.
Okay, sir. Is it 8985706364, sir?
8985 70 63
64.
Okay.
Okay, sir. Thank you.
Okay. All the best.
Thank you, sir.
Okay. Shall I leave?
Thank you, sir.
Okay, ma. Thank you very much. Shall I leave, ma?
Okay.
Okay. All the best. We will discuss tumor immunology tomorrow. Okay.
Okay, sir.